full paper presentation given by Marwan Abdellah, and he's going to talk about NeuroMorphoVis. And he needs some time to prepare the setup.
We still have 3 minutes. I don't know if we can start now, but I don't know if you wish to wait.
Okay, so we're going to wait 2 more minutes to synchronize with the other sessions, I've been told.
Okay, thank you very much, Mark. So, happy to be here. BioVis, I actually wait every year to come here. So this year I'm going to present to you a new tool, novel tool called NeuroMorphoVis, a collaborative framework for analysis and visualization of neuronal morphology skeletons reconstructed from microscopy stacks. Again, long title, but I do like it. So before actually starting explaining what NeuroMorphoVis is doing, I have to explain the reason why did we develop it. So in the Blueprint project, we build a brain, but in silico. We try to simulate a brain and reverse engineer the mammalian brain in order to understand its function and structure. And with that, actually, we have a long pipeline, an iterative one that starts from the wet lab doing some experimentation, and then we try to build tissue models. And from the tissue models, we perform some simulations in supercomputers, and then we do some analysis. And finally, we do some validation, visual analysis and validation using some visualization tool. So we basically go to the lab, and then we start tracing the neuronal morphologies, and we reconstruct a microscopy stack. And from this microscopy stack, we have a digital model, a morphology of a neuron. And that's the basis of our entire simulation, this little piece of neuron. And then actually, we do that for diverse type of morphologies, and we end up having different types of morphological types called M types, and these are 55 different ones we have reconstructed. So we use these basic morphologies actually, and then we mix and match them, and we try to put them somehow in the building unit of our simulation, which is called the neocortical column. And then we go to the lab and we measure voltage traces, and we reconstructed what's called an E-type or electrophysiological type. And from this 11 E-types, we combine and map the function, the E-types, on the structure, and we build up finally a simulation like this. That's a digital piece of a 2-week-old rat simulating the function of it. With the blue colors are more hyperpolarized, and the red ones are depolarized. So that's the first use case actually. Every little piece here, it's a neuron, it's a morphology. Imagine if this morphology is broken, is wrong. then of course there's a a question here. Then what about the accuracy of the results? The results won't be accurate anymore. So we have to make sure that each morphology, each respective morphology is right. Another kind of experiment, we do what's called an in silico optical experiment by building tissue models, tagging them virtually with fluorescent dyes, and then we try to image them as if we can image them under the microscope in the wet lab. We have already published this in BioVis 2015, and in 2016 actually we extended the simulation for highly scattering tissue, and then in 2017 we presented a framework to reconstruct large-scale volumetric models. So that's fine, but then what's the common factor? It's all about morphologies. We have millions of morphologies actually that we use for our simulations. So what's the issue here? The issue comes as follows, that scientists coming to us telling us, okay, we have many morphologies, but we don't know if these morphologies are okay or not. So we need some tool that would allow us to do morphology visualization. Some other scientists come to us like, okay, fine, but we need also another tool that would allow us doing the analysis, visual analysis. And also, we would like to get a tool that would allow us repair the broken morphologies. Like if you go on neuromorpho.org, you have, I don't know, 100,000 morphologies, something, and then like 90% of them are broken because of the tracing artifacts they used to have in the wet lab. And then the SOMATA are simply represented by very, very simple primitives like sphere or cube or something like this. So we need to have a valid way of doing the SOMATA reconstruction. And also in order to visualize the simulations, we need to have mesh models. So we need to have a tool that would allow us generating mesh models. And potentially we need also another tool that would allow us generating volumes, huge volumes that would give us the capability of building in silico optical study. And also got recently a request that we need to have a tool that would allow us just simply generating media for our papers. We just like put some bunch of neurons and then we end up like having a very, very nice image that would allow us like increasing the acceptance rate or the probability of our paper getting accepted. So we thought actually, okay, we have a bunch of requests that we might need to build individual tool for each thing, but that won't be a good thing. But rather, we should build one tool that would allow us to make everything in a collaborative fashion, doing everything just in a single user interface. And from this idea, we came up with NeuroMorphoVis. It's a Blender-based tool that simply does everything here. And here I will just try to give a glimpse of, the functionality of NeuroMorphoVis, but of course, the time won't allow us to explore everything here. So I'll simply start as follows. We have morphology database. And from this morphology database, we import the morphologies from different standardized file formats, and then we try to repair the morphologies. We have different sets of automated filters that would allow us repairing the morphology. And once we repair them, we simply put them back in the database. So even if we keep having new and new morphology in the loop, we just repair them and then we put them back in the database to ensure that we have no broken morphologies. Once we fix the broken morphologies, we simply do some analysis. We generate some graphs to see exactly distribution of the branching across certain arbor, or the number of bifurcation, trifurcations, the distribution of the radii across the arbor, and so on and so forth. So we have to also provide a single click that would allow us to generate a series of graphs for hundreds of thousands of morphologies, just like that, in an automated way. And then we can also generate some visual analytics by having different types of reconstruction or rendering techniques that would allow us actually seeing the different aspects of the morphology. I'll come to that later. And then from the fixed morphology, we generate different types of surface meshes, and also we export them in different file formats. And finally, we generate from these mesh models volumetric models, and we annotate or tag them with the actual optical properties of the cortical tissue and also with any with the spectroscopic properties of any fluorescent dyes that tag them. So this is basically the workflow of the framework. Just it's a simple one, but with every little box here, like lots of other modules that are actually integrated there. And this is the software stack of the tool. So we have a command line interface and user interface. So the user can basically go to Blender and then just launch it and then load the plugin and then start using the tool. Or with command line interface that can be like plugged into like a web interface or something for large scale generation. And then we have NeuroMorphoVis modules that are based on some core API with some Blender functionality, and at the end we use the Python API of Blender with optional dependencies, our PBB software, and also H5Py and Slurm for distributing the data on clusters. So I just quickly go through the morphology visual analysis capabilities. Normally, when I just check any kind of paper, and people would like to demonstrate different morphologies, just use black colors like this. And if they try to make it sexy a bit, though, they actually tag the axons or color the axons in blue and the dendrites in red. And that's it. But this is actually something very basic. What we need is to have something like this, a tool that would allow us to tag each component, including the SOMATA, the apical, basal, and even oblique dendrites and the axons with different colors or even different shaders and lighting conditions. So we have actually added like this module to allow us tagging the different components of the morphologies. And also we can control, like select each component in each arbor and map it to what we can see in the input file, whether it's an SWC file or H5 file, so that we can do that mapping and then we can figure out what the errors. And also we have shipped the tool with different reconstruction methods with like different radii and things like that in order to improve, enhance the visual appearance of the reconstructed morphology. So for the reconstruction methods, for example, we have added 7 methods, actually 6 with one that can vary the color, that would allow us see the morphology as different pieces of like segments connected together with different colors or the same color in A and B, and also showing the morphology as disconnected, like a piece of disconnected sections, and then showing each arbor as a disconnected polyline. And in F, it's the morphology is one piece, and potentially in G, we have the soma reconstructed on a physically plausible basis. So that's the full morphology after being repaired and reconstructed the soma in a way that would allow us to see the structure of the morphology itself. We also added a feature that would allow us controlling the geometry. This is a full morphology, but you can barely see it. So if someone would like to get the global view of the morphology, he won't be able to do it. So we have added actually some features to manually control the geometry and increase the radii of each branch so that we can figure out the global view. And also, we can control the branching order. So in A, we can see that the axon is highly, like it's making flutter, so it's barely, it's very tough to us actually to see the other components, the apical and basal, so we can control the branching order. And we also added the arbor selectivity feature, which would allow us actually getting only the components we need. So in A, we have the full morphology. In B, we ignored the basal dendrites, and in C, we ignored the apical dendrites so that you can see the shape of the SOMATA in blue are totally different. And finally, we added also some sketching methods that allow us, removing the little like zigzag thing in the original morphology in A and making them tapered to be right, and also adding some sort of artistic method of drawing the morphologies. Okay, so the next thing is SOMATA reconstruction. Normally, we have the SOMATA represented by spheres or cubes or cylinders, and this is not true because what we can see in the ASCII files is basically the profile is represented by a 2D profile like this. In order to get a 3D profile like this, we need an advanced optical microscope, and the majority of the morphologists do not, they haven't been reconstructed by such microscope. So we have actually added a module in order to, that would allow us reconstructing the 3D shape of the SOMATA on a physically plausible basis using Hooke's law. And we have made that for already 55 different morphologies we have had in the blue brain. And also we have added capability to change the parameters of the SOMATA, and then we can compare actually what we can reconstruct from the tool and what we can get from a 3D microscope already, and then we can validate the reconstructed SOMATA. The next thing is the mesh reconstruction. And with that, actually, we take an input morphology skeleton, and then we reconstruct the mesh. We have different meshing algorithms. So these are just for different morphologies. And as you can see here, we have two basic algorithms. We have already enhanced that in the latest release into 4. So we have the extrusion-based meshing and the piecewise watertight meshing. And also we preserve the mesh quality by using vertex smoothing operations, and we reduce the number of polygons with tessellation operation, but we still preserve the quality, so we measure actually the error and then we report them. And then the last thing is the volume reconstruction module, which would allow us like building volumetric models of each neuronal morphology. And then we can scale that from one single neuron to a group of pyramidal cells to a full cortical column and even to a cortical slice that has more than 200,000 neurons. And also we have reconstructed these volumes, and we rendered them using our in silico bright field microscope. So this is actually a simulation of the bright field imaging of the neuronal tissue, and that's already an ISBC Art and Science poster downstairs. And that's another poster. It's an in silico brainbow that simulates the fluorescence microscopy of a piece of tissue that's tagged with different fluorescent dyes. And the last thing is the scientific media generation. So people coming to us, we need media for our papers. So we have actually added a module here that would allow the scientists just directly loading the morphology and getting some nice media like this. So these images have been created with NeuroMorphoVis in a direct way. So all you have to do is just give a bunch of morphologies with a configuration file, and then you can generate some images like this. That's a spectrum of pyramidal cells showing the diversity. If we just have a close-up, it looks like this. And also we have used the tool in order to reconstruct GLIA, which is almost similar in terms of structure to the morphology. And these images have been just produced like in a few minutes. So the last thing here is the open sourcing. The tool is supposed to be open source. There's already a GitHub repository, but if you just go there, you'll find the README file and nothing else. And then if you open this README file, you see that it's not that apparent that the tool should be open by, the initially should be available by May 1st. Actually, the tool would be open source. We are just waiting for the university to confirm the open sourcing process, no more. But if you're interested in using the tool, just send us an email and we'll gladly send you the code that you can use it, but we get a promise that you don't disseminate it. So last thing I have to say is the tool is really, it's full of tons of options that can do so many things. Of course, you still need to read the Bible for the manual in order to understand how to use the tool itself. But at the end, if you just try to play with it a little bit, you can get very, very nice analysis and media out of it. And with that, I would like to acknowledge the ISMB for the travel fellowship actually we have received to present this paper. So thank you very much.
Thank you very much.
Time.
Questions? Is there a question? I can start with one. So you talked about collaboration. Do you mean co-located collaboration or remote collaboration?
Both, because basically we have like, I don't know, 6, 7 teams. Each team is concerned with one certain workflow in our pipeline, and then like the morphology analysis guys are coming to the visualization guys. You see, our morphology looks bad, so we need to figure out what's the issue. So the visualization guys, they have to take the morphology, and then they have to see what's the issue, and then they have to get back to them. But with this tool, basically, we can just, both teams, they do have they can sit together and then they can investigate what's wrong, or they can just simply share the morphology and then they can generate the reports, and then the Viz guys and the morphology analysis guys talk to each other, and then it's going to be way easier actually to handle that. Yeah.
Thank you.
A question from an outsider. So first of all, good talk, great talk. The question from the software perspective, I see that you have lots of features, and from a user, I think that sometimes lots of features can be a blessing, but also can be a curse.
Yes.
Just for a beginner, he or she may get lost in using these features. Is there any way that you can keep the features, but have an easier interface for them to access these features? Thank you.
Yeah, that's a very good question, actually. I've received a lot of comments about this. So basically, this is the tool. Well, it's not shown here. So this is the tool. When you just load the tool, you get these toolboxes. None of them is open, so that I don't really make a shock for the user when he's trying to use the tool. But if you just click here, and then this drop-down menu, it shows you the specific toolbox that you are interested in. So if you go to the morphology analysis, you just have this. And then you just simply toggle it back, and then if you'd like to go to the SOMATA toolbox, you have these options and nothing else. And same thing for the morphology toolbox. You have blah, 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 and that's it. So you just need to get to be aware of what kind of toolbox that you're looking for, and then you have to use it. But this is Blender. This is the way Blender is designed. And I think Blender is a very, very nice tool, but you just need to spend like a couple of days actually trying to, see how does it work. That's it.
Okay, thank you very much. Thank you again.